Here we are talking about the challenges linked to the evolution and transformation of the world of parenteral medicine and the implication of Annex 1 guidelines. So the new revision of Annex 1 implies the implementation of contamination control strategies. And this is the most important and challenging aspect of the quality management in manufacturing of injectables nowadays. Contamination control strategies plays a vital role in the management and control of contamination from microbial, endotoxin, pyrogen, and particulate derived from sterile manufacturing process in order to ensure the performance and quality of the product in the drug manufacturing process. And this involves all the elements that take part to the process. That means raw materials control, equipment, process design, and also management of outsourced activity. And as far as primary packaging is concerned, Annex 1 has multiple ramifications on manufacturers related to new requirements with primary packaging components, such as elastomeric components, specifically stoppers, plungers, which are involved in contamination control strategies documentation. The monitoring and reduction of particulates and microbial contaminants from primary package components is one of the main aspects in ensuring that the finished product is free from contamination. And in aseptic manufacturing, the components are required to be cleaned using validated processes to ensure that particle, endotoxin, and pyrogen and biobarrier contamination is adequately controlled. And the new revision strongly recommends also the use of barrier technologies such as restricted access barrier systems, REBs, and isolators in order to minimize the microbial contamination that originates from human intervention. And depending on the drug manufacturer existing infrastructures, they may be required to install new equipment and or make extensive adjustments or in order to comply with new guidelines. Should the manufacturer choose not to adopt these technologies, the alternative method needs to be justified. And container closure integrity is a part of contamination control strategy documentation, and the manufacturer should ensure package integrity and sterility at various product life cycle stage. This includes validation of container closure combination used with drug products to ensure that the configurations used can maintain package integrity and sterility until the end of its shelf life. And these factors may contribute to the efforts toward reduction of contamination, as primary package components have an impact on the overall contamination control strategy. And the second aspect we will talk briefly is about some examples on how emergent formulation approach can meet clinical needs by tailoring therapy to the individual patients. And there is a great deal surrounding the concept of personalized medicine. Personalized medicines is rooted in the belief that since individuals possess nuance and unique characteristics at the molecular, physiological, environmental exposure, and behavioral levels, they may need to have interventions provided to them for their diseases they possess that are tailored to this nuance and unique characteristics. And this belief has been verified to some degree through the application of emerging technologies such as DNA sequencing, proteomic imaging protocols, and wireless health monitoring devices, which have revealed a great inter-individual variation in disease processes. Actually, the personalized medicine is a big deal, but there are a number of challenges associated with it, especially with respect to obtaining their approval for routine use from various regulatory agencies. In addition, they have many issues associated with broad acceptance of personalized medicines on the part of different healthcare stakeholders, such as physicians, healthcare executives, insurance companies, and patients. And among these challenges, there are the proof that personalized medicine strategies is outperforming traditional medicine, even considering that this kind of medicine is very expensive. The most known, that I think nowadays, the main type of personalized medicine is CAR-T cell transplant therapy for certain type of cancer and mutation-specific medicines, and such and also immunotherapy in cancer therapy. So, about the most advanced characterization techniques in the context of interaction between modified release delivery systems and primary container, should take into account that the interaction between modified release delivery systems and the primary container is crucial for ensuring the efficacy, stability, and safety of pharmaceutical products. And here we can recognize different characterization techniques that are important in this context. That means that you can't approach this type of characterization only with a technique, but you need to use different characterization techniques and to combine them. And the most useful techniques are spectroscopic techniques, including infrared spectroscopy, Raman spectroscopy, and nuclear magnetic resonance, microscopy, such as advanced microscopy techniques, including scanning electron microscopy, transmission electron microscopy, and atomic force microscopy, thermal analysis, such as differential scanning, calorimetry, and thermogravimetric analysis that are commonly used to study thermal behavior of materials, and are important when a material such as a primary packaging material comes in contact with stressing conditions such as sterilization condition with heat, and mechanical testing to test the mechanical properties of the material, chemical compatibility studies, and also particle characterization to highlight particle leaching. And if we need to characterize the interaction between a modified release delivery system and the primary container, we need to combine, as said, different techniques in order to have a picture, a complete picture of the problem, of the issue, or of the behavior, we say, of the packaging material.